Bismillah ar-Rahman ar-Rahim. My name is Dr. Khalid Abul Bashir. I welcome you all to my YouTube channel, the channel that I call uh, Molecular Genetics. In this channel, I will be giving lectures on genetics, molecular genetics, medical genetics, population genetics, and molecular biology. I am actually a molecular biologist. Uh, I welcome you all to this channel. I hope you find it useful what I give in these lectures, and I will start my lecture today by talking about Mendelian genetics. Uh, an introduction to genetics. This lecture will be about Mendelian genetics, and then my name and the objectives of this lecture. Okay, the Mendelian genetics. Today I will start also talking about Mendel's laws of inheritance. So in this lecture, I will be talking about who's Gregor Johann Mendel and his life, his experiments, what made him succeed when others failed, and laws of dominance and laws of segregation, law of dominance and segregation, so known as the monohybrid ratio of 3:1. And then we go to the law of independent assortment, or dihybrid ratio of 9 to 3 to 3 to 1. And after that, I will talk about incomplete dominance of a ratio 1:2:1 ratio. And I will give a short discussion about brief short talk about the Mendelian paradox. This is Gregor Jan Mendel, but before talking about him and his life and his experiments, his laws, I would love to just give a brief historical overview about the science of heredity and genetics. I normally divide historically the science of heredity and, uh, and genetics into three historical eras. And the first era, I love to call it pre-Mendelian era, and then the Mendelian era, and post-Mendelian era. The pre-Mendelian era starts very early in time, and then comes until Mendel. Then the Mendelian era, life of Mendel and his work, and then after that the post-Mendelian era, which starts by the year 1900 with the rediscovery of Mendel's laws. In the pre-Mendelian era, we can say that early attempts in the field of heredity date back to 6000 BC, when historical evidence showed that Babylonians and Chinese did so many work on, for example, Babylonian tried to prepare pedigrees for their horses, and they did some sort of cross pollination of date palms, and Chinese farmers tried to improve rice varieties by selection for desirable characters. So all these attempts, they were very early in history, 6000 and 5000 BC, and then came the Greeks in 3000 something. Of course, the Greeks, they were interested in the human reproduction and sex determination and the likeness of families and this. So Greeks, the famous Aristotle actually did some work on what are the contribution of the father and the mother to the embryo, and who does what and who doesn't. And then the Romans and Arabs also did some work on plant and animal breeding and pedigrees. So all these historical trials were just trial without any scientific background, or they followed no scientific approach. It was not until the 17th century that a truly scientific approach began to be taken, when scientists started to focus scientifically, following a scientific approach, scientific tools, and applying some principles to decipher, understand, and explain some of the phenomena, genetics, and abnormalities. Uh, um, um, Many scientists studied human genetic disease. For example, in 18th century, a scientist uh, known as Pierre Louis de Maupertuis studied the polydactyly phenomena, that is an additional digit in the hand or the foot. So in a certain family, and he explained how this actually runs in a family, and it gets passed from one generation to another. And then John Dalton, he was a doctor who suffered from color blindness, and few of his relatives also suffered the same thing, and he tried to study and explain the heredity behind, and how this heredity behind color blindness, and how this disease or abnormality get passed from one generation to another. Also, we have some scientist called Inalles Martin, who studied hereditary blindness, another form of blindness, in the early 19th century. And George Huntington, George Huntington, who studied a neurological disease which later took his name, and we have the Huntington disease till today, which is a genetic disease. And also William Osler, who reported number of new hereditary conditions. And we have Joseph Adams, who had some reports on Friedreich ataxia, and he tried to classify the Friedreich's ataxia into familial and genetics, and he had some work on this field. John Hay also, who established a full details on the inheritance of hemophilia. And all those scientists and another, they have their contributions in the field of genetics. And finally, in the era before Mendel, we have the heavyweight scientist Charles Darwin, with his major work on Origin of Species, and the other book, Variation of Animals and Plants Under Domestication. All these works, and also we cannot ignore Francis Galton, the cousin of Charles Darwin, and so many other scientists and biologists who did their share on the field of genetics. The problem is all those people did this without prior knowledge of principles of genetics, and the only one who started to focus and postulate some sort of hypothesis to explain how these diseases and how these abnormalities actually, and how these traits and characters get passed from one generation to another, is uh, Gregor Johann Mendel, our scientist, our father of genetics. And he was born, I think I will talk about Gregor John Mendel. I'll talk about who he is. I'll talk also about when he was born, and where and how. He is a father of genetics, as I said, and he was born in a place called Heisendorf. Heisendorf in district Silesia, Styria at that time. Nowadays, it is known as Czech Republic. He was a monk. He went for the Augustinian monastery as a monk for some times in 1843, and then he became a monk at that monastery in Brno, also in the Czech Republic. And by 1847, he became a priest, and from there he was sent to University of Vienna in 1851 to study natural science. during the years of his studies in Vienna University of Vienna, actually he polished his skills, experimental skills and analytical skills, so that uh, these skills actually uh, helped him focusing on his experiments later on. And after that, he returned back. He returned after he finished his years at University of Vienna. He returned back to his monastery, where he was given a limited space in the garden of his monastery, and he carried his pea hybridization experiments at that space for 8 years, from 1856 until 1864. And what he actually did, Mendel, he did some experiment using a, a garden pea plant, also known as Pisum sativum, and he continued his experiments for 8 years, after which he formulated his laws of inheritance. During his lifetime, the work of Mendel did not actually attracted much of attention in the scientific society. So it was not until after his death. It was not till after his 35 years after his death. He actually died. And was presented before he dies. He presented his paper and his results in the Natural History Society at Brunn in 1865, and then his paper was published after his death in 1901. But in 1900, after his death, 35 years after his death, three uh, of very renowned scientists, De Vries and Karl Correns and Tushmak, these three scientists actually they published their work, and fortunately they had similar results as John Mendel experiment, and they published these three uh, papers of these three scientists on different journals independently, and the publication of this work, which is similar, which as I said, similar to John Mendel's, Gregor Mendel's, is known in the scientific world as a rediscovery of Mendel's law. So uh, after the rediscovery of Mendel's law in the 1900, now Mendel's work became to be appreciated, and after his death, actually. Okay, what made Mendel succeed when others? Of course, there were so many others at Mendel's times, Mendel's time, and before Mendel, they did the similar experiments, plant hybridization, plant breeding, and crossing, but they did not reach the same conclusion as Mendel. What made him succeed when others failed is a very big question. Mendel was a very fortunate and far-sighted person, scientist, I would say, in which, in what? He was fortunate and far-sighted in his choice of plant, the plant that he did the experiment with, and in his choice of the characters that he studied. He actually studied 7 characters in the garden pea, Pisum sativum. As I said, he studied these 7 characters in that plant, and fortunately for Mendel and for us, of course, that he chose that plant in particular. Why? Because the garden pea was easy to grow. This is what made him choose the plant, and easy to grow and breed, and also they produce a very large number of seeds, which allowed him to do the mathematical analysis. And the plant also was not susceptible to random fertilization by stray pollen grains, so it was very good for the experiment. And also there were numerous at that time, there were numerous pure strain readily available of the garden pea. So these and another along with other factors, that it is an annual plant and it has a very well defined characteristics that can be easily studied, and can be easily also grown across the plant, and it has a perfect flower, very big flowers, and they are as I said available in pure lines. All these made it very nice and very helpful for Mendel to go on with his experiments. Mendel chose 7 characters, as I said. Unfortunately for him, that these characters were later proven to be actually located. The genes responsible for these 7 characters were actually proven to be located in 7 different chromosomes. And also, for the good of Mendel's and for being a very fortunate person, fortunate and very scientific as well, very far-sighted person. Also, the plant itself, the garden pea, also is a haploid, a 7 haploid chromosome, a plant that has only 7 haploid chromosomes. That's why Mendel obtained independent assortment in F2 generation. We'll talk about this when we talk about the laws of inheritance. He obtained independent assortment in F2 generation without linkage producing any new recombination and any contamination. The linkage, if the plant was diploid or something, so a linkage would have changed some characters. So Mendel wouldn't have reached the same conclusion as he did with the garden pea. This is very fortunate, and at that time he did not know about the genetic makeup of the plant. He did not know that the plant was a 7 chromosome haploid plant, and of course he did not know that the 7 characteristics that he chosen for his experiment were actually located in 7 different chromosomes separately. So what Mendel did, first, Mendel chose a pure line plant. He actually worked to get a pure line plant. Pure line means a plant or pure strain. Pure strain means that a plant, for example, if we are talking about a long plant and a dwarf, long plant and short plant, he actually grew that long plant for some generation to ensure that only the progeny, the offspring, what that plant will give, the plant that will be produced from that long plant will be all long plants. So he grew that long plant and self-fertilized that plant for so many generations to ensure that this plant will carry only the character of tallness. On the other hand, he also did the same thing with the short plant, the dwarf plant. So he grew a long plant and a short plant for so many generations to ensure that this will give him a pure line plant, pure line. So now he got a pure line plant for all the characters he studied, for long plant, short plant, and for long plant, short plants, and for we'll talk about the 7 characters: the color of the seed, the form of the seed, the form of the bulb, the uh, the color of the flowers. All these characters Mendel studied. He did the same with all of them. So he took the garden pea and self-fertilized it for so many generations to get a pure line. So what he did? The problem with the garden pea that is it is a self-fertilized plant. That the plant, the male sex organs and female sex organs are present in the same flower. So what he did, as I said, the plant has a very big flowers. So he opened the flowers and carefully removed the stamens. He carefully removed the stamens so that he emasculated the plant. He removed the male sex organs from the plants, and then he pollinated the stigmas carefully using a very fine brush with pollens from other plants. So what he did, took a plant with some characters, opened the flower, took the stamens, all the stamens, emasculating the plant, and then he pollinated the the stigmas with a pollen from certain from other plant of a certain characters. That means if he had a long plant, he opened the flower of the long plant, took the stamens from the long plant, and took pollens from the short plant, and then he did the crossing, he did the fertilization between a long plant and a short plant, so that he ensures the plant does not self-fertilize. So he did this, and after that he put a bag on the pollinated flowers so that to avoid contamination from other pollen parents. At a time he selected only one pair of contrasting characters. For example, this is one of the good thing that shows good attitude that Mendel followed in his experiment. He took one character at a time. He studied one character at a time. For example, the color of the flower first. We said that he studied 7 characters, but at a time he studied one character. After he finishes with that character, he goes for the second and the third, and after that he studied 2 characters together, and 3, and so on. So first he studied one character having one trait, one character having a contrasting characters. So he selected a pair of contrasting characters: tall and short, red and white, flattened and constricted, and all a pair of contrasting characters. So, and in his experiment, he gave the dominant. I will talk about dominant and recessive. He gave the dominant character a capital letter, and the recessive one he gave it a small letter. So first, let us talk about what are the characters that he chose for his plant to study. First, form of the seed forms, how the seed look, either round seeds or wrinkled, either round or wrinkled. He gave the round seed letters, 2 letters actually for each trait, for each character, because we know that a character is coded in a gene, and we know that every animal or plant or human, they have a gene. One comes for the same character, one comes from we have two genes, one comes from father and the other one comes from the mother. So he gave the character two letters. That means one letter coming from the father and the other one coming from the mother. So for the first character he studied was form of seeds, and we have two contrasting characters in guarding P. Either round seeds, and he gave that round seed RR capital, and or wrinkled seeds, and he gave the wrinkled seeds small r and small r. The other character he studied, the first character we said that the form of the seed, shape of the seed, and then color of the cotyledons. Color of the cotyledons, either yellow cotyledons or green cotyledons. The yellow cotyledons, he gave the character yellow. That means the yellow is a dominant, and the round seeds are dominant. Yellow cotyledons are dominant, so he gave the yellow cotyledons a capital Y. While green cotyledons, he gave them two small y's. The third character he studied was color of seed coats and flowers. Color of seed coats and flowers. Either gray or white. Either gray or white. So the gray took the letters G, two G's, capital G's, and the white took the small G's. The fourth character he studied was the form of pod, either inflated pod or constricted pod, and he gave the inflated pod the capital F's, and constricted pod he gave them the small f. Then the color of pod, form of pod, then color of pod. This is the fifth. The color of pod, either yellow or green, gave the yellow capital, two capital Y, and the green two small Y's. Position of the flower, either axial or terminal. The axial flowers gave them capital X, and the terminal flowers he gave them small x. Length of the stem, either tall stems or dwarf stem, capital T for the tall and small t for the dwarf. So this is what he did. He chose a pure line plant, one for example for the red flower and the other one for the white flower, or gray and white, gray and yellow. Excuse me, sorry. Either flower gray or white flower, and then he crossed these two pure line plants, and he observed for the progeny of that crossing between the two contrasting plants. So now we talk about the law of dominance, first law of Mendel's. This is what he did. Mendel first, he took one plant carrying a pure character and the other carrying a contrasting character to the other one, and opened the flower, took the stamens, removed them, then pollinated the plant from the other one, the pollen from the other one, and then covered the flower with a bag and waited for the plant to grow, and then took the seed and grow them to see the character, to study the characters of the result of that crossing. So the first law of the first Mendel's inheritance law is known as law of dominance and law of segregation. Law of dominance and law of segregation. In some references you find that they put these two laws differently, like they give first law of dominance and then they say law of segregation, another law, and then the third one is law of independent assortment, third law. But here I will talk about law of dominance and law of segregation as one law. I'll call it first law of uh, first Mendel law of inheritance. Okay. Uh, in a cross of parents that are pure and contrasting traits, taking two plants having contrasting traits, and he hybridized those two plants. Uh, Only one form of the trait will appear in the next generation. Let me first stop here to explain one point. That we have two terminologies. We have something called phenotypes and we have a genotype. A phenotype is, if you are talking about a plant, talking about a plant, for example, how the plant look for example, the flower. If the flower of that plant is red, then the shape, the color of that flower is known as the phenotype of that flower. So phenotype means the appearance of a trait, while genotype, the other term, genotype means the genetic makeup of responsible of that phenotype. Phenotype is the appearance of a character, and the genotype is the genetic makeup, the shape of the genes that are responsible for that character, that phenotype. So this is what Mendel did. Actually, he took a plant, he crossed a true breeding plant, for example, tall plant. For example, in this example, he took a tall plant, he gave a T, capital T, to indicate a tall plant, and then a short plant with a small t. So now we have two plants. This one plant is a tall plant and the other one is a short plant. The tall plant will have two t's and the short plant will have two t's, small t's, and these are capital T's. Now, when he crossed these two plants, one tall and the other short, that means tall plant will have two genes, capital, two dominant genes, and the short will have two small genes, that's two small t's. So these t's, when the plant form the gametes of the plant, one t will go in one gamete, the other t will go in the other gamete. Here also in the short plant, small t will go in one gamete and the other small t will go in the other gamete. So now when he crossed the tall and the short, what will happen? He will get one from the tall and one from the short. So the progeny will be T capital and T small. The second one also, the other T of the tall will fertilize the other T of the short, small t. So we'll have two plants, one carrying capital T and small t, and the other one carrying capital T and small t. Let me put it again in another word. For example, if we cross the T and the T, what we will get? This is a tall plant and the short plant. The offspring, the plant that is produced by this crossing, will be carrying capital T and small t. So this plant will carry capital T and small t. This is the genotype of the plant. So will be capital T and small t, the genotype, the genes. One gene will be dominant, the other gene will be recessive, small, capital. So the plant produced from this crossing will carry capital T and small t. This is genotypically. What about the phenotype? The phenotype, dominant when we call this trait is a dominant trait. A dominant trait is a trait that appears when a dominant gene is present along with a recessive gene. For example, in this cross, this is not shown now. And what is shown is a tall plant crossed with a short plant. The result will be capital T and small t. Now, the plant that carry the capital T and small t actually carries both contrasting genes. One gene responsible for tallness, the other one responsible for the shortness. So what the plant will be? Will it be tall or short? What Mendel found, he found that when we cross a dominant plant, a plant that carrying a dominant gene and having a dominant trait, with a plant that carrying a recessive gene with a recessive trait, when we cross these two plants together, the resulting plant, the progeny that is produced from this cross, will always be a tall plant. So the phenotype, and this one will be the F1, first generation. The first generation, when we take two pure breed, two pure line plants having contrasting traits, when we cross them together, the first plant produced from this crossing, from this hybridization, the first plant will produce from this hybridization are known as the F1 generation, first generation of the cross. So the F1 generation, all the first one generation will be showing a tallness. The plant will be tall. This is why we call the tall trait, this trait, as a dominant, and this one as a recessive. So what is dominant and what is recessive? When Mendel crossed true breeding tall plant with a true breeding dwarf short plant, the F1 hybrid, the F1 generation, always looked like the tall parent. F1 generation always looked tall. And when a true breeding red flower, now if we took a plant that is having a red flower with another one having a white flower plant, and we cross these two plants together, the progeny, the F1 generation, will always be a red flowered plant. So Mendel called the character, the trait that appears in the F1 generation dominant, and the character that is suppressed in the F1 generation, masked in the F1 generation, as recessive. So now what is dominant and what is recessive? Dominant and recessive are when we cross two true breeding plants or animals that are having a contrasting traits, having contrasting characters. When we cross them together, the trait, the character that appears in the first generation, F1 generation, is always known as dominant character. And the character that disappears, masked, suppressed from the first generation, from F1, is always known as the recessive character. So this is what he did. After he did the cross between the two true breeding plants, one tall and one short, and he got the T capital and T small, and the plant was the plant produced were all tall plants. He went a step ahead by doing a self-hybridization. Self-hybridization means he took one plant from F1 generation and crossed that plant with another plant from the F1 generation. So this is called selfing or self-hybridization or self-crossing. He crossed an F1 plant with an F1 plant from F1 generation, and the other plant from F1 generation. We can put it in another words. That is, we know that, as I said, the green pea is a self-fertilizing plant. So he fertilized the plant, he self-fertilized that plant, or he crossed two different plants from F1 generation. It will be the same result. So what will happen? This is the gene, this is the genetic makeup of the first plant, and this is the genetic makeup of the second plant. So why this is put, why we put this as T and this as T and this as T and this as, why we separate them? Because we know that in the plant will carry these genes. One gene will be T capital and one gene will be T small. But when the gametes are formed, when the plant start forming the gametes, we know that, for example, in human, that sperms have one of the two sex chromosomes. Either one sperm is having either Y or X. The other sperm will have either Y or X. That means, but the body cell, somatic cell, will have both chromosomes. This is, I think I need to to give a lecture about mitosis and meiosis and cell division to make this understandable. But I'll just talk about it briefly. The human, a man, will carry in his body, the cells of his body will carry 46 chromosomes, 22 pairs of autosomes, 22 pairs of autosomes, and one pair of sex chromosomes. This pair of sex chromosome will either be an X or X and Y. X and X will give a female, and X and Y will give a male. So when a man, the man will carry an X and Y. So his cells will carry one chromosome X and one chromosome Y. When that man forms his sperms, the cell will divide into two. So the sperm is produced from one cell, one mature cell. Spermatogenesis, the process of spermatogenesis. Sperm is formed from one cell, one somatic cell. So that somatic cell will divide, resulting into two sperms. Actually, it is more than two, but let's say it is two sperms. The two sperms, one will carry the X chromosome, the other one will carry the Y chromosome. So X chromosome and Y chromosome in each sperm. Similar thing will happen in the plant. The plant that carry T capital and T small, when it forms its gametes, one sperm, for example, if it's correct to say, the sperm, one sex cell will have, one gamete will have a T chromosome that's, or a gene that's a dominant gene, and one recessive gene. So it will divide like this. This is a cell when the plant form the gamete. T will go in one gamete and the T small will go in the other gamete. The other plant also, T will go in one gamete and the other T will go in the other gamete. So this box shows us that when fertilization, when we cross these two plants, what are the chances? What are the odds? What are the possible progenies from this cross? Either one gamete, the gamete that carry this capital T, fertilize the gamete that carry this capital T, and the plant that produce from this crossing, from this hybridization, will be carrying both dominant genes. Or the gamete that carry this small t will fertilize the other gamete that carry the capital T, and the plant produced will be carrying a capital T and a small t. That means dominant gene and recessive gene. Okay, or this gamete, the gamete that carry this T, capital T, will fertilize the gamete that carry this T. So it happens randomly. One of this option will happen. So either this one will fertilize this one, and the resulting plant will be, the resulting progeny will be carrying a T capital and T small. The other one, the other possibility, that is a gamete carrying a small t will fertilize the gamete carrying a small t, and the resulting plant will be carrying both small t's. So these are the possibilities. So now this is a genetic makeup. This is a genotype. So when we cross the F1 generation with another F1 generation, what will happen? The possibilities genetically that the genotype of the offspring of the progenies will be one carrying both dominant genes and two carrying a heterozygous. Heterozygous means two different genes. Two of the plants will be a heterozygous, one will be homozygous. This is a homozygous because both are the same thing. One homozygous dominant and two heterozygous and one homozygous recessive. This is the produced plant. The progenies will be the genotype of the progenies. One is a homozygous dominant, two heterozygous, and one homozygous recessive. So this is a genotype. So it will, the genotype, the genotypic ratio will be 1 and 2, 1 is to 2, those 2 and 1. So the TT will have a possibility of 25%. Two heterozygous will have the possibility of 50%. And the homozygous recessive will have the possibility of probability of a 25%. This is about the genotypic part. What about the phenotypic? How the plant will look? How the produced plant will look? Phenotypically, the ratio will be 3:1. Why? Because T capital and T small, the plant will be tall. This is not. And if you remember when I talked about the F1 generation, now we are talking about F2. This is F2 generation. Because F1 we said a long plant carrying TT crossed with a short plant carrying small t, and then the produced plants will all be carrying T capital T small. This is genotypically. And phenotypically, the F1 generation genotypically will all carry T capital and T small. While the phenotypically all plant in F1 generation will be tall. Now in F2 generation, how we get F2 generation? When we cross a plant from one F1 generation with another plant from one F1 generation. And this will produce three types of plant will be, 3 plants will be tall and 1 plant will be short, because the plant that carry 2 dominant genes for tallness will be tall, and this heterozygous will be tall because the dominant gene will dominate the recessive one, and here as well the dominant gene will also dominate the recessive one, so the trait will be tall, the plant will be tall, this also will be tall, and this also will be tall, while the plant that carries 2 small t's, or the recessive genotype, will be short. So phenotypically we will have 3 tall plants and 1 short plant. Genotypically we'll have 1:2:1. So this is called the monohybrid cross. Cross. So the first law, the law of dominance, that is when we cross a true breeding plant. When we cross a true breeding plant, carrying a character with another true breeding plant carrying a carrying a contrasting character, all the plants in F1 generation will look like one of the two parents. So the trait that appears in the F1 generation is known as a dominant trait, while the one that is suppressed will be called as F2 will be called as recessive trait. And from the results of Mendel, from these results, he actually concluded that from F2 generation, concluded that the two alleles, this allele means form, will have this is when we talk about a gene, some genes are having two alleles, like this one. The gene of tallness, the gene responsible of plant tallness, is having a two allele. Two alleles means two forms, two shapes. One shape is capital T, the other shape is a small t. One shape is dominant, the tall, the other one is the short, the recessive. So this is allele. Allele means the shape of a gene or a shape of a character. So what Mendel concluded? He concluded that the traits, these traits, the tall and the short, segregate for the two alleles. Those are segregate. They do not mix. In this animal, they do not mix together. That means if they mix together, the produced animal will be a medium animal, medium height animal. So we are talking about plants. The if they mix together, that means the produced plant will be a medium height plant. But and when we cross this medium height plant with another medium height plant, the result will be a medium height plant, because two medium height plants will give us a medium plant. So this is not the case. What was the case? The case was crossing the F1 generation with another F1 generation. Instead of giving us a medium height plant, this gave us a 3 long plant and 1 short plant. That means these traits actually segregated, they separated, they were not mixed together, they were not mixed. If they were mixed together, that means they will remain like that. But then they segregated. Every trait went to a gamete. This trait, this T, this gene went to one gamete, the other one went to the other gamete. So he concluded that traits segregate for the two alleles. For these two alleles, the traits segregate for the two alleles, the T capital and the T small, where in F2 generation. In F2 generation, this, like when we are going to F2, this will go to one gamete and this will go to another gamete. The other plant, one will go in another one gamete and the other one will go to other gamete. So this is a segregation. The F2 generation in a 3:1 phenotypic ratio, resulting, this segregation will result in a 3:1 ratio. And these characters segregate but do not contaminate each other. They do not contaminate, do not mix with each other. They do not blend with each other. So this is called the law of segregation. So now we talked about the law of dominance and the law of segregation. The law of dominance means when we cross two true breeding plants carrying two contrasting traits, when we crossbreed these two, the resulting generation, the F1 generation, will show one of the contrasting trait of the parent plants, and this trait that is shown by the F1 generation is known as the dominant trait, while the one that is suppressed or disappeared means or called the recessive trait. Law of segregation, he said, when we cross the F1 generation, F1 plant with another F1 plant, filial one plant with another filial one plant, and those heterozygous, those hybrids, the traits in those hybrids will segregate in a way that when they mixed again to form the F2 generation, the ratio, the phenotypic ratio will be 3:1, 3 of the dominant and 1 of the recessive, and that means the traits in the F1 generation did not mix, they did not blend or contaminate each other. Instead, they segregated to give us in the F2 generation a 3:1 phenotypic ratio and a 1:2:1 genotypic ratio. This is known as a law of segregation. So now we know the law of dominance and law of segregation. This is the first law of first Mendelian law of inheritance. Now we talk about the second law of inheritance, which is also known as the law of independent assortment. Uh, the law of independent assortment, it is stated like this: when two or more characteristics, now in the first experiment, what we talked about, the first law of inheritance, law of dominance and law of segregation, we only studied, or Mendel studied, one character, that is tall and short, the tallness of a plant, that is one character, with two contrasting alleles, that's two alleles, two shapes, that character, one character, but two shapes. In case of law of independent assortment, Mendel actually took things one step ahead. Instead of studying one character, he studied two contrasting characters. So how the law of independent assortment goes? It goes like this: when two or more characteristics are inherited, when we study the inheritance of two or more characters, individual hereditary factor, that is genes, individual genes assort. Assort means assort independently. They assort independently during gamete formation. Now we are talking about two genes. Are they inherited together, or they assort? Everyone is inherited independently. They assort independently, independent of each other. We are talking about two genes together in the same plant, not two different genes in two different plants. Now we are talking in one plant, we are taking two genes or three or four. When we study the inheritance of such genes, two genes or more, two characteristics or more, uh, the genes responsible of these characteristics will assort independently during the gamete production, giving different traits, giving different characteristics, and equal opportunity of occurring together. Let's go and explain this in this slide. In this case, Mendel took a pair of contrasting. Instead of taking one pair of contrasting characters, here he took two pairs of contrasting characters at a time. And then he crossed them with their recessive characters. Now, let's see here. This is, he took a two pair of contrasting characters. He studied the color and shape of the seed, color and shape, color and form, seed form. Now, color, one character is the color, the other one is the form of the seed. Color can either be yellow or green. The plant will be either yellow, or the seed will be either yellow or green. Now the shape or the form of the seed. The form either round, in both cases, or wrinkled. So the plant, so green and round, green. Green is a dominant. Green is a dominant character. Yellow is a recessive character. Round is a dominant character. Wrinkled is a recessive character. So he took one plant that's having a round and green, so dominant, both are dominant. Round, this is a dominant for the shape, and green, dominant for the color. He took this plant and crossed it with a plant that is carrying a seed of a wrinkled shape and a yellow color. So we have a recessive plant for two characters and a dominant plant for two characters. He crossed those two characters, and now I'm sorry. The yellow is the dominant character. Yellow is the dominant character. Yellow is the dominant, Y, capital Y, and the green is recessive. Green is the recessive. So he took one yellow and wrinkled with one round and green and crossed them together. So this plant carries one gene, one trait as a recessive, the wrinkle shape, and one gene as a dominant, which is the yellowish color. The other plant carries seeds with a round shape, which is a dominant shape, and a green color, which is a recessive color for the seed. When he crossed both those plants, what will he get in the F1 generation? We said in the first law of inheritance, we said that the progenies in F1 generation will always show the dominant characters. The dominant character. Now we talk about the dominant character. The yellow is the dominant and round is the dominant. So the F1 generation all will be round and yellow. F1 generation will all the plants produced by this crossing will be yellow and round. After that, in the first law, if you remember, the first law, we just took one character. For example, the wrinkle, the shape, and the roundness, the round shape. Then he crossed them, and he found the without considering the other character. This is for the first law, because he studied just one single character of contrasting alleles. Here he took both one seed carrying a dominant and the other and a recessive. The other also carries one dominant and one recessive. And when he crossed them together in the F1 generation, the seed in the F1 generation were all yellow and round. So this is F1 generation as usual, as we expect from the law of dominance. That is the F1 generation will always show the dominant characters. So this is true. Now what he did, we did the same thing as we did in the F1 in the first law of inheritance. That is, we self the F1 generation. That means we crossed the F1 generation with another plant from F1 generation. We crossed a plant that carrying a yellow round seed with a plant that carries a yellow round seed, and now we see how the possibilities are. These are the gametes for male plant, and these are the gametes for the female plant. For example, say this is a male and this is a female. Now the gametes are like this, and the possibilities. So this one will always carry capital R and small r. The F1 generation will have a capital R coming from here and a small r coming from here. and capital Y coming from here, and small y coming from here. This is for the color. So the round shape is due to this capital R here, and the yellow color is due to this Y. So when we segregate the traits in the gametes here, when the gametes are formed in F1, this one, it will be either the gamete will carry capital R and capital Y. This gamete will either carry capital R and capital Y, or carrying a capital R and small y. One gamete will carry capital R and capital Y, the other gamete will carry capital R and small y, a third gamete will carry a small r and small y, small r and small y, the fourth and the last one will carry small r and capital Y. So this is the gamete for this one. Now similar thing will happen in this one also. Capital R and capital Y, capital R and small y, small r and small y, and small r and capital Y. So now if this gamete fertilized with this one, what are the result will be? What is the result will be? The result will be capital R and capital R, capital Y and capital Y. So this plant will be a round and yellow colored plant because it carries all dominant genes. What about this one? If this capital R and small y fertilized with this capital R and capital Y, the result will be capital R and capital R, and capital Y and small y, and also the plant will be round and yellow. Why? Because round because of the dominant genes or dominant trait, and yellow because the presence of one dominant trait will suppress the other one. So the trait that will appear is of this capital trait, capital Y, that is the yellow color. Similar one here. The gamete will this one will be fertilized with this one. So when we fertilize this with this will happen randomly actually. So the possibility of this and this will match together. That means this capital R and small r, and capital Y and small y. So also this will be round and yellow. Round because of the presence of the dominant gene, and yellow because of the presence of the capital Y, the dominant gene. The fourth one also capital R and small capital R and capital Y match or fertilize the small r and small y. This is the same like this one. So capital R and small r, and capital Y and small y, capital Y and capital Y. So this will be also round and yellow because the R capital and this is dominant. So this also will be. So we'll have 4 round and Ys. The other possibilities, this capital R and capital Y with capital R and small y. R and R capital and capital Y and small y also will be round and yellow. Okay. R capital and Y capital with R capital and Y capital. R and R will be round, will give us a round shape, and small y will give us the green shape. This is because Y and Y, this is a recessive, so this will give the green color, which is a recessive trait. Small r and small y will give us R with capital R and small y. This will give us capital R and small r will give us the round shape because of the presence of the R, and small y and small y will give us the recessive trait, that is the green trait. Small y and capital R with capital R and small y. This is capital R and small r will give us the round shape, and small y with the capital Y will give us the yellow shape color. So this will be yellow. Now this one, small y, small r and small y with capital and capital will give us round and yellow, capital and small with both small, r capital with r small, round shape, yellow small with yellow small will give us a green color. Small r, small y, small r and small y, this is will give us wrinkled, r and r will give us the wrinkled form, shape, and small y and small y will give us the green color. Uh, Fertilizing or matching between this gametes and this gametes will give us R and R. R and R will give us the wrinkle shape, and capital Y and small y will give us the yellow shape, yellow color due to the presence of this dominant yellow genes. Small r and small and capital Y with capital Y and capital R, this will give us a yellow round. The round due to the presence of this capital R, and yellow due to the presence of capital I, capital Y, and capital Y. So this will be yellow and round. Capital R and small y with small r and capital Y, a round shape due to the presence of the R, yellow color due to the presence of the capital Y from here. Small and small y and r with a small r and capital Y will give us a wrinkled shape due to the presence of R and R, small and small, recessive, and capital will give the presence of this capital gene, capital Y, this dominant gene will give us the yellow color. Small r and small r will give us the wrinkled shape, capital Y and capital Y will give us the yellow color. So what is the ratio, the phenotypic ratio? In this case, when we talk about two characters together, taken together, this will give us a 9, 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11. Round, yellow and round, 1, 2, 3, 4, 5, 6, 7, 8, 9. Yellow and round. This is the dominant character, yellow and round. And then we have half dominant and half recessive. Half dominant means round, and half recessive green. This will be 3, 1, 2, 3. Again, half dominant, half recessive, yellow color dominant, wrinkled recessive, also will be 3, 1, 2, 3. And all recessive, green and wrinkled, this will be 1. So the ratio will be, the hybrid ratio will always be 9 to 3 to 3 to 1. Excuse me. This ratio gave us 4 traits, and this means when we cross this F1 generation and F1 generation, they'll give us a one phenotype. This one phenotype, this is F1 generation, and in which in this generation, in this F1 generation, both the traits, all the 4 traits, all the traits means R small, R capital, Y and Y, all are present without blending. They are present together, side by side in this F1. All these possibilities are present in F1 generation, side by side, without contamination, without blending with each other. So in F2 generation, the 4 traits are sorted independently. They do not go alongwise. That means small r it's not fixed to small y. It can go small r with small y, small r with a small y, or small r with capital Y. So they are sorted independently. They are distributed independently, independent of each other. Means a capital R, it's not always fixed with capital Y, because we find capital R with small y. So it is a possibility. So that means when we take 2 characters or more and study them together, the inheritance of those 2 and more characters, and how they get inherited, in F1 generation we find the F1 plants are all showing the dominant traits, but in F2 generation, we will find the ratio of 9 to 3 to 3 to 1. That means those two traits, they are not always fixed together and inherited together, but instead they are actually assorted. They are found in F1 generation side by side. When they get inherited in F2 generation, they are assorted independently of each other. So this phenomena is called the second law of inheritance, and also called the law of independent assortment. The law of independent assortment, that means when we consider two characters together, and when we study those two characters and cross them with a plant or animal having a contrasting characters of these two characters, and in F1 generation, F1 will always show the progeny in F1 will always show the dominant characters, while in F2 generation, the results indicate that the four traits are actually found side by side in F1 generation, and when we get past to the F2 generation, they get assorted independently of each other. This is, and they always give us the a 9 to 3 to 3 to 1 ratio. This is called the dihybrid ratio. These are the laws of inheritance postulated by Mendel. Now we come to the incomplete dominance. Incomplete dominance, it's a situation in which one allele, now if we just go back to the first law of inheritance, the law of dominance, it is always like that. When we cross a plant or animal that's carrying a character with a plant and animal that's carrying a contrasting character, we expect in F1 generation the characters that appears are the one of these two characters in the parents, the dominant one. So in case of incomplete dominance, we have something called incomplete dominance. That is a situation, a genetic situation in which one allele does not completely dominate the other one. In case when one allele does not dominate the other allele, so the result in the F1 generation will be a new phenotype. When we get a new phenotype, in such a case, this is a flower of snapdragon plant. Snapdragon carries gene that give us incomplete dominance. Flower of snapdragon, one flower is red, the other one is a white flower. When we cross these two plants together, we expect either a red color or a white color. But what we get in that, this is a case actually soon after rediscovery of Mendel's law. Mendel did not get such result after Mendel died and his laws were rediscovered after 1900. Scientists started getting results different from the classical 3 to 1 ratio. That is in F1 generation, when we talked about the law of dominance, 3 to 1 classical ratio. They did not get that ratio. They got something new, totally new. 1 is to 2 is to 1. This is called incomplete dominance. So in F1, they did not get 1 into 2 into 1. They got this in F2. But in F1, they got a completely new shape. For this example, crossing a red flowered plant with a white flowered plant, instead of getting either red or white, we got a pink. This indicates that the dominant allele did not actually completely dominate the recessive one. So that means dominant got expressed and the recessive also got expressed at the same time. So they got a mixed color, which is a pink color. In this case, in such a case, the phenomena is known as an incomplete dominance. And just to make sure what we see in front of us is an incomplete dominance, the scientists actually did a cross test to just make sure this is uh, a new thing. It's not the usual uh, uh, 3 to 1 ratio, monohybrid ratio. So what they did, they took this pink color plant, this pink flower plant, and crossed it with the red one. When they crossed this one with this one, they crossed both this pink and red. What they got was something strange. They got a red plant and a pink plant with a ratio of 1 to 1. So they got one red and one white. That means if this one, if red was a dominant gene, and when we cross it with this one, we should have got at least 3 red and 1 pink. But this is not a completely dominant gene, the red one. So that's why we got a red one, we got a pink one. We got one pink and one red in a ratio of 1 to 1. Okay, maybe this is the recessive. Let's go with a white plant, white flower plant. So they did the same thing. They took one plant with a pink flower and crossed it with a plant with a white flower, and what they got actually the same result as when they did with the red flower. They got a one white flower plant and one pink flower plant in a ratio of 1:1. So they called such a situation an incomplete dominance. This should not be confused with another phenomena, that is a codominance. We have incomplete dominance and codominance. In codominance, both genes are considered to be dominant. So part of the plant will be showing the trait of the first dominant, and the second one will be showing the trait of the other dominant. For example, if we took sheep, we crossed sheep, a black one with a white one. If we consider that a black color in a sheep is a dominant one and a white color is the dominant one, when we cross these two together, a white sheep with a black sheep, then what we'll get? We'll get a sheep having a black spots and white spots. It will not be brown or something mixed color, a gray color or something. No, it will be part of the sheep will be showing black, completely black. Part of it will show completely white. So in such a case, this is called a codominance. But when we get a mixture of two, in such a case, we call this an incomplete dominance. Now, finally, I will conclude my lecture today with something called Mendelian paradox. Mendelian paradox. Uh, in 1936, a statistician called R.A. Fisher, a very renowned statistician, he stated that based on he carried out chi-square analysis on Mendel's results. He just wanted to make sure if Mendel has correctly analyzed his result. He did a chi-square analysis, and he concluded that Mendel data was too good to be true. So this was a shock to the scientific world, because Fisher actually made it bluntly that Mendel's data was too good to be true statistically. So he believed that either Mendel falsified his observation, or he modified the data so that adjusted the observation so that he get these results. And at the same time, in the scientific world, they know that based on the characters of Mendel, and he's a religious man, and so the characters actually, his character suggests that Mendel's character suggests that he was unlikely to engage in either a deliberate falsification of his data or an unconscious his scientific background and his religious character all showed that or indicated or give us a hint that he was unlikely to engage in either a deliberate falsification of his data or unconscious adjustment of his observation. So now we are in a dilemma. In one part, a statistician is saying that the data is too good to be true, and in another hand, the man is too good. The man is too honest to falsify his data or uh, adjust his observations so that he get the result. And there are so many papers published discussing this paradox, the Mendelian paradox, about the how can we just using a statistical tools to make sure if the data is falsified or not, is it possible or not, or can we make sure that the Mendel's data was not falsified based only on his characters and what we believe about him. So this is known as Mendelian paradox. With that, with just this brief talk, I would conclude my lecture today. I hope that it will be useful for everyone in the field of genetics. And it is an introductory lecture, Mendelian inheritance, Mendelian genetics. Next lecture, I look forward to seeing you in the next lecture. And thank you so much.